Now let us discuss about osmosis. Osmosis means the movement of the solvent, that is water molecules, across a semi-permeable membrane from a low solute concentration to an area of high solute concentration is known as osmosis. Here must have to be present the semi-permeable membrane, because the semi-permeable membrane only allows the solvent molecule and it prevents the entry of solute molecule. Now, the exact amount of pressure that is required to stop osmosis is known as osmotic pressure. In this diagram, we see there is equal amount of solvent and solute molecules which is separated by a semi-permeable membrane. Then subsequently here we see there is only movement of the solvent molecule, but solute molecules remain the same. This movement is known as osmosis. What is the importance of this, physiological importance or significance of this osmosis? Absorption from the intestine, continuous osmotic exchange is going on between blood, body fluid, tissue fluid, cells and lymph, regulation of urine formation, reabsorption of cerebrospinal fluid. Where osmosis is important, as we know, in case of kidney for reabsorption, in case of intestine for absorption, in case of capillary bed for continuous exchange. Now let us discuss about active transport. As we know, the active transport is divided into primary active transport and secondary active transport. Secondary active transport is again divided into co-transport and counter-transport. Active transport: the movement of the ions and molecules across the membrane against a concentration and electrical gradient from a lower concentration to a higher concentration with an active expenditure of energy with the help of carrier protein is known as active transport. When we say the definition of active transport, we must have to mention all these points. This active transport is also known as uphill transport. It is like swimming in the opposite direction of water in the flow of a river. What are the criteria of active transport? The movement of molecules against gradient, that is concentration or electrochemical gradient. It requires carrier protein. It requires energy in the form of ATP. Active transport must have require energy in the form of ATP and transport proteins against the concentration gradient. The transport is again divided into primary active transport and secondary active transport. As we know, the primary active transport, ATP directly required for the function of the carriers. This ATP comes from the breakdown of adenosine triphosphate, and there is conformational change in the carrier protein occurs to move the ions to the other side. The example of primary active transport is sodium potassium pump. Now let us discuss about very important topics of sodium potassium pump, which is also known as sodium potassium ATPase pump or sodium potassium adenosine triphosphate pump. What happens here? It is a primary active transport process that pumps sodium ions outward through the cell membrane of all the cell, and at the same time pumps potassium ion from outside to inside the cell against their concentration gradient with an active expenditure of energy by the help of carrier protein. What happens there? Sodium potassium pump transport 3 sodium ions out of the cell and at the same time 2 potassium ion into the cell. Sodium potassium pump, its carrier protein is a complex of two separate globular protein. One is the larger subunit known as alpha subunit, whose molecular weight is 1 lakh, and the smaller one is beta subunit, whose molecular weight is 55,000. It has 3 receptor sites for binding of sodium on the portion of the protein that protrude to the inside of the cell, and 2 receptor sites for potassium to the outside. Inside the portion of this protein near the sodium binding site, there is sodium potassium ATPase activity. As we see the figure, this is a cell membrane, this is the outside cell, inside cell, and this is the carrier protein, that is globular protein, which has 2 portion, larger portion and smaller portion. What happens here? 3 sodium from inside it goes to the outside by binding with the carrier protein, and at the same time 2 potassium from outside comes into the inside of the cell membrane with the help of the ATP. Sodium potassium pump is always called an electrogenic pump. Why? Because it carries an electrical potential across the cell membrane. Sodium potassium moves three ions outside the cell and at the same time two potassium ions inside the cell. Thus, in every pump, the cell loses one positively charged ion from the cell that creates positivity outside the cell. So, inside the cell, there is deficient of positive ion which leads to negativity inside the cell. That's why the sodium potassium pump is called electrogenic pump. Now let us the functions of sodium potassium pump. Sodium potassium pump is responsible for generation of membrane potential. It maintains the cell volume. It is the basis of nerve function, that is transmitting nerve signals throughout the nervous system. It creates electrogenicity. Aldosterone, insulin, and thyroid hormone increases the pump activity. On the contrary, dopamine, cardiac glycosides inhibits the pump activity. Now, secondary active transport. The secondary active transport, where the energy is derived secondarily from the energy that has been stored in the form of ionic concentration differences of secondary molecular or ionic substances between two sides of the cell membrane. It is divided into co-transport and counter-transport. Co-transport: in case of secondary active transport, two substances are moved across the cell membrane by binding with the same carrier protein in the same direction. It is also known as symport. Example is sodium glucose co-transport, sodium amino acid co-transport. As we see here, sodium is binding with the sodium binding site and goes inside the cell. At the same time, glucose binding with the glucose binding site and goes in the same direction within the cell membrane. Now, counter transport. It is that opposite of the co-transport. It is also known as antiport. Here, the substance move across the membrane by binding the same carrier protein in opposite direction. The example is sodium calcium counter transport, sodium hydrogen counter transport. As we see here, this is the outside cell, this is the inside cell. Sodium binding with the sodium binding point and goes inside the cell. At the same time, with same carrier protein, calcium bind from inside the cell and goes to the outside. Now, vesicular transport or macromolecular transport, which is divided into exocytosis and endocytosis. Endocytosis is again divided into pinocytosis and phagocytosis. Now, exocytosis. Exo means out. What happens there? Exocytosis is the reverse of endocytosis. It requires energy. It means when a substance must have to go outside the cell, it forms a vesicle, then it fuses with the cell membrane and releases contents to the outside the cell. By definition, it is the process by which the cell releases the macromolecules or contents of secretory vesicles out of the cell of the membrane is known as exocytosis. It may be waste products, proteins, hormones, or some other products of secretion. Now, endocytosis. Endocytosis means comes inside. Endocytosis is the taking of molecules or particles by invagination of the cell membrane by forming a vesicle. It also requires energy. This is the process by which very large particles enter into the cell by a specialized function of the cell membrane is known as endocytosis. What happens here? These molecules have to be entered within the cell. Then the cell membrane become invaginate. Then it form a vesicle. Then release the vesicle inside the cell. Endocytosis. Now, endocytosis is divided pinocytosis and phagocytosis. Pinocytosis is also known as cell drinking. What happens there? Ingestion of minute particles that are in solution and not visible under microscope is called pinocytosis. Example, ingestion of protein, vitamin, etc. Phagocytosis, also known as cell eating, it is the process by which bacteria, dead cells, or tissue debris are engulfed by the cells such as tissue macrophage and some of the white blood cells. What are the steps of phagocytosis? Recognition and attachment, engulfment, killing, and degradation. Now, this is a very important topic, transcytosis. It is also known as vesicle tracking or vesicular transport or cytophemesis. Transcytosis is the transport mechanism in which an extracellular macromolecule enters through one side of the cell, migrates across the cytoplasm of the cell, and exits through other side of the cell. Example: movement of some proteins across the endothelial cell. Many pathogens like HIV virus, that is human immunodeficiency virus, also transported by this mechanism. How it occurs: this is endocytosis one side followed by exocytosis other side. As we see this picture, what happened, the substance that enter into the cell through endocytosis, they form a vesicle, they enter within the cell, then it goes outside the cell through exocytosis. Significance of the transcytosis: it plays an important role in selectively transporting of substance between two environments across the cell membrane. Thank you.